Hi, everyone. Welcome to our seminar series on the cerebellum. So today we have as our guest Jing-Ning Zhu, who received his PhD and his bachelor's degree from Nanjing University in China, where he remained for his career. He's currently a professor of neurobiology and vice dean for the School of Life Sciences at Nanjing University. He's also the president of the Jiangsu Provincial Society for Physiological Sciences in China. His research has made major contributions to understanding the link between the cerebellum and the hypothalamus in such tasks as control of feeding. Today, however, we will hear from his work on the role of the cerebellum in translating exercise into alleviation of anxiety. Thank you so much for taking the time, Jing-Ning.
Yeah, thank you. Good morning, everyone. Actually, at this time in China, it's very late at night. Firstly, I want to thank Reza for your very kind invitation, and I am very pleased to have this opportunity to present our recent study at Johns Hopkins Cerebellum Seminars. And today, I want to talk about the role of the cerebellum in motor and emotional integration. So movement is very important to humans and animals, since it is the basis for the execution of behaviors, including the feeding behavior, and the courtship behavior, and some behaviors associated with motor learning. The central motor structures responsible for the control for the movement are organized hierarchically from the motor planning and programming and motor initiation, motor execution to the motor coordination. Among them, the cerebellum is the largest subcortical brain structures, plays an important role in the motor coordination, motor balance, and regulation of muscle tone. Cerebellar lesions or cerebellar circuit dysfunction will result in ataxias. For example, our previous studies found that the downregulation of the CRF in the red inferior olive neurons, this is the sole origin of the climbing fibers for the cerebellum, leads to gait disturbance and motor incoordination, whereas injection of the CRF into the cerebellum will rescue the ataxic motor deficits. In fact, an intact behavior not only requires the external somatic movement, but also involves various internal non-somatic or non-motor components, such as the visceral activities, emotions, sleep and wakefulness, and even language and cognition. Therefore, when facing challenges in the external and internal environments, we need to coordinate and integrate somatic motor and non-motor responses to generate appropriate behaviors. So the question is, what are the neural mechanisms responsible for this somatic, non-somatic integration? So in recent years, we have done a serious study on these questions and find that there are direct bidirectional circuits bridging the motor structures, including the cerebellum vestibular nucleus and the basal ganglia, and the high center for regulating visceral and autonomic activities, the hypothalamus. We have reviewed that the hypothalamus may directly modulate the neuronal activity of the cerebellum and basal ganglia, and consequently regulates the motor coordination, motor initiation, and even psychomotor activities. In turn, the cerebellum may also innervate the hypothalamic feeding-related neurons and cardiovascular related neurons to actively participate in the metabolic and cardiovascular regulation via the direct cerebello-hypothalamic projections. And in this talk, I will focus on the cerebellar regulation on the emotions. So the mental disorders, including anxiety disorders, depression, and bipolar disorders, affect a great amount of population these days. We all know the physical exercise can improve the mood. Whether walking in the park or running in the gym can help us to recover quickly from a stressful event. Accumulating prospective cohort studies also support that physical exercise, such as 45 minutes each time and 3 to 5 times a week, and 2.5 brisk walks a week, or 12 weeks exercises, confers protection against the depression and anxiety. However, little is known about the underlying mechanism. Most current studies have focused on the general interactions between the periphery of our body and our nervous systems. And they find that the peripheral tissues or cells such as fat, liver, and muscles can release various transcriptional factors or cytokines into the brain through the blood-brain barrier during movement, thereby regulating the neuronal activity and to regulate the emotions or cognition of the brain. But what we think is that both the coordination of the movement and the control of the emotions are two separate yet highly interconnected functions of the brain. So the motor actions not only constitute an important component and a physiological basis for the expression of emotions, but can also improve our mental health. So is it possible that there are neural substrates between the central motor system and the emotional system that mediate the motor-emotional integration? In 1998, Professor Shadmehr and his colleagues report cases of the cerebellar cognitive affective syndrome. These patients have an isolated cerebellar injury, but manifest disturbance of execution functions, impaired spatial cognition, language deficits, and inappropriate affect and even personality changes. More and more recent studies have focused on the non-somatic or non-motor functions of the cerebellum. The cerebellar granule cells have been found to encode reward signals, and the cerebellar nucleus can send direct projections to the VTA. This is the rewarding center in the brain. And this projection may be actively involved in regulating the social behavior. So we know the cerebellum is closely related to the autism. But what for the emotions and anxiety? Neuroimaging studies also hint at a possible relationship between the cerebellum and anxiety. Anxiety patients have greater gray matter of the cerebellum and show greater cerebellar reactivity to the normal phase and scenes. For the cerebellar circuitry, although the Purkinje cells are the principal and the projection neurons of the cerebellar cortex, actually the cerebellar nuclei, including the fastigial interpositors and the dentate nuclei, are the final integrated node and the outputs of the cerebellum. So our working hypothesis is whether the cerebellar nuclei can innervate and modulate the limbic system to mediate the motor-triggered mood regulation. So first, we cooperated with the psychiatrist at Nanjing Brain Hospital to collect the resting state fMRI data from the patients with bipolar disorder, in which we know the anxiety is the most prevalent comorbid diagnosis. We use the Pearson correlation analysis and find that the Hamilton anxiety rating scale, the HAMA score, but not the depression score and the mania score, show the trend of the negative correlation with the cerebello-amygdala functional connectivity in the BD patients. Next, we use the viral neural circuit tracing to dissect whether the presence of direct cerebello-amygdala projections in the rat. By using the anterior grade virus tracer, we find that the cerebellar nucleus can send direct projections to the amygdala, particularly in the lateral central amygdala, but not in the BLA and very less in the CEM. By using the retrograde tracing, we find that the neurons projection to the CEL distribute all the three cerebellar nuclei, but especially most of them are distributed in the dentate nucleus. And then we employ the monosynaptic rabies virus with the fMost to tracing the direct projections from the cerebellum nuclei to the CEL. And this is the reconstruction of the three projections from the dentate nuclei neurons to the CEL amygdala neurons in the mice. So the distribution patterns of the DN neurons projecting to the CEL in the mice is very similar to that of the rats. So this means this projection may be conservate in the conservate the projections. Then what's this what's the function of this cerebellar amygdala projections, and especially its roles in the regulation of anxiety? So we use the alleviate plus maze and the light dark box to assess the level of anxiety of the animals. And we find that after running in the rotarod at a constant speed, the animals shows less anxiety levels. This means the constant rotarod running can elevate the anxiety. And then we use the GCamp7 to label the amygdala neurons And combined with the optogenetics manipulation, we find that the amygdala neurons can be activated during the animals running in the rotarod. And interestingly, this amygdala neurons response in the running can also be activated by the opto activation of the DN neurons. When we use the opto inhibition to the DN neurons, the amygdala neurons response or activated in the running at the rotarod, their response is blocked. Will be blocked in the running. So this means the the cerebellar amygdala projections can directly activated in the during the movement, and these projections can directly activated the amygdala neurons. Then we use the resting state fMRI to determine whether there are other the important structures related to the anxiety maybe have also maybe activated in the running. So we find that only the amygdala, but not the MPFC or NAC or hippocampus can be their functional connectivity between the cerebellum and them did not have significantly change after the running, but only the functional connectivity between the cerebellar nuclei and the amygdala shows significantly change after the rotarod running. Then we employ the patch clamp recording with the optogenetics manipulation to determine the electrophysiological properties of these projections. And we find these projections are glutamatergic, and these projections is monosynaptic. Since the after the running, and actually the movement can induce lasting alleviation of the anxiety. So we also test whether this projection activation can induce synaptic plasticity. So after high frequency stimulation of the terminals of the amygdala cerebellar afferents, so we find the amygdala neurons can show RTP. And we also use the biosighting to lab the neurons we recorded in the patch clamp recordings, and find these neurons are PKCδ positive neurons. It has been reported that the PKCδ neurons in the CEL can bidirectionally regulate the anxiety. So then we use the opto activation of the DN-CEL projections, and find it can induce an activation of the CEL PKCδ positive neurons. And both the opto activation and the chemo activation of the cerebello-amygdala projections can induce an improvement of the anxiety. On the other hand, chemo inhibition of these projections can block the motor-induced alleviation. So next question is, what types of exercises are more effective in improving the mood? So using the behavior test on the rotarod, the simplest different types of the movement is the accelerating rotarod. So we test the accelerating rotarod running and compare their effect on the anxiety with the effect of the constant rotarod running on the anxiety. And we find that accelerating rotarod running can improve the stress, the CUMS-induced anxiety more effectively than the constant rotarod running. So what's the mechanism mediate the more effective improvement effect of the accelerating rotarod? So because in our previous studies, we find that the orexinergic neurons in the hypothalamus can send direct projections to the vestibular nuclei. This is actually the nuclei in the brainstem to control the muscle tone. The orexin actually firstly was identified for its function to promote the feeding behaviors, but the later studies find that orexin deficiency may induce narcolepsy, cataplexy. Actually, the cataplexy is a sudden loss of muscle tone, and this is actually is a motor deficits. So this makes us to think the function of the orexinergic system in motor control. And we find that the hypothalamic orexinergic neurons can directly innervate the vestibular nuclei. And very interestingly, these projections did not play an important role in the routine movements or general movements, but it plays a key position when animal facing the motor challenge, such as accelerating rotarod or inclined balance beam, but not the horizontal balance beam. So this means these projections may be involved in the motor challenge. So we think whether these projections may be responsible for the effect of the accelerating rotarod, or means the challenge movement. Then by combining the tracing and also the C-force immunostaining, we find that accelerating but not the constant rotarod running can induce an activation of the hypothalamic orexin neurons projection to the cerebellum. And consistently, we also find an elevation of orexin levels when animal running at accelerating rotarod in the cerebellum. So we know that the orexinergic system can project almost all structures in the brain, including the amygdala. So what we ask the question is whether this the orexinergic neurons projection to the amygdala can is the same population with the orexinergic ones projection to the cerebellum. So we use the CTB and the FG to retrograde labeling the orexinergic neurons in the hypothalamus, and find that the orexinergic neurons projection to the amygdala and to the cerebellar nucleus are totally separate, means the different population of the neurons. But this remains a question that whether the orexinergic neurons projecting to the amygdala can be activated during the movement. So we also use the retrograde tracing and the C-force and the C-force immunostaining to find that neither the accelerating rotarod running nor the constant rotarod running can induce activation of the orexinergic neurons projecting to the amygdala. And the orexin levels in the CEL did not change in the constant and accelerating rotarod. So this means the orexinergic neurons projecting to the amygdala did not be activated during the movement. So for specifically manipulate the orexinergic projections to the cerebellum, so we generated orexin pre-rat and use the chemogenetics to selectively inhibit the projections from the hypothalamus to the cerebellum. And we find that chemogenetic inhibition of these orexinergic projections suppress the motor-induced the elevation of the anxiety. And then we use the pharmacological techniques, actually by using the agonist and antagonist for the orexin receptors to further test the orexin projections effect. And we find that blockage the orexin 2 receptors in the cerebellum, or using the lentivirus to knock down the orexin 2 receptor expression in the cerebellar nucleus, can animate the improvement effect of the anxiety of the challenger movement. And using the patch clamp recordings in the cerebellum slices, we find that the orexin excites the glutamatergic projection neurons rather than GABAergic interneurons in the cerebellar nuclei. And we also test the ionic mechanisms underlying the excitatory effect of the orexin on the cerebellar nuclear neurons. And we find that both activation of the sodium-calcium exchangers and the closure of the inward rectified potassium channels mediated the excitatory effect of orexin on the DN-glutamatergic projection neurons. Then we use the optogenetics manipulation combined with the multiple channel recordings in the cerebellum DN to test whether the cerebellar nucleus projection to the amygdala can be activated during the accelerating movement, and whether the activation of the DN neurons can be blocked by the opto inhibition of the orexinergic fibers. And we find that during the constant movement, the neurons' projection to the amygdala can be, their firing rate can be increased. And during the accelerating rotarod, their firing rate is gradually increased during the accelerating rotarod. And when we selective inhibition of the hypothalamic orexinergic inputs, and the firing rates, the level of the firing rate of the DN neurons recover to the previous level during the general movement. That means the on the constant rotarod running. So this means the during the accelerating rotarod, the hypothalamic orexinergic neurons can be recruited to give more excitation on the cerebellar DN neurons, and this excitation maybe provide additional driver to activate the cerebellar amygdala projections. So we also use the orexin 2 receptor antagonist, the CTS, to test whether it can block the effect of the accelerating rotarod on the activation of the PKCδ positive neurons in the amygdala. And we find that blockage of the orexin 2 receptors in the cerebellar nuclei can effectively inhibit the motor-induced activation of the cerebellar amygdala projections. So at the last, we use the optogenetics combined with the chemogenetics to manipulate these three neurons circuits. And this experiment is confirmed that this hypothalamic cerebello and amygdala projections can mediated the motor triggered alleviation of the stress induced anxiety. So the last question we want to address is what our findings can make some suggestions or implications for the clinical. So we know the movement is actually an effective method, maybe used in the clinical for elevate the anxiety. So we test the different accelerations in the rotarod and find that the more accelerating rotarod can induce a more effective improvement effect on the alleviation of the anxiety. And even we only use the optogenetics to activation the activity of the cerebellar nuclear neurons, and we can use the different frequency to activate the level of the activity of the cerebellar nuclear neurons to their activity level during the constant and the accelerating rotarod running. And we find that the higher frequency stimulation can induce more elevated or amelioration effect of the movement on the anxiety. So this means in the future, maybe we can use some neuromodulation techniques such as the TMS or other methods to manipulate the cerebellar activity to maybe we can improve some mood disorders. So summary, in this study, actually we find these three neurons circuits. In fact, during the general movement, the projections from the cerebellum to the amygdala can be activated. And this activation of the projections can make effective amelioration effect on the anxiety. And but if we using the challenge movement, we can recruit the orexinergic neurons in the hypothalamus to provide an extra driving force for the cerebellar amygdala circuits to give a more amelioration effect on the relief of the anxiety. So this study may highlight the potential use of the challenge strategies to optimize the exercise therapy or cerebellar neuromodulation manipulation for the mood disorders. So this study has just published on the Neuron as a cover story, and Professor Kodakar also write previews for our studies. So I wish to thank my collaborators, Professor Chris Zhu at Erasmus MC, and Professor Wang Fei and the Nanjing Brain Hospital, and the Di Jun and Professor Jianjun Wang. I also this work is actually the first author is the Professor Zhang Xiaoyang, and also the PhD my PhD student Wenxia, Shen Liping, and Ji Miaojing. So I also wish to thank the following organizations, including the MOST and the National Natural Science Foundation of China, and the Ministry of the Education, and the Natural Science Foundation of the Jiangsu Province, China, for their financial support. So thank you very much.
Oh, Jing-Ning, that was just amazing. Thank you so much for that presentation. I'll start the questions, and if anybody has questions, please just go ahead and let us know, and we will come to you and allow you to ask questions. But I was just wondering, in the studies that you showed, the rotarod, it's it's a kind of a stressful thing, I think, for mice, because usually the rotarod, it's unusual, I suppose. And I wonder, is when have you trained them multiple times on the rotarod, and is the effect that you see on their willingness to go explore, does it does it have diminishing returns after repetitive exposure to the rotarod, or is it something that is sustained despite repetition?
Yeah. Thank you, Reza. This is a very important and good questions. Actually, we note these issues, and in our test paradigm. So we actually we this the constant speed we use in the rotarod, not only in the constant rotarod and accelerating rotarod, we test their anxiety behaviors after the motor training. But we find that there was no significant changes in their anxiety levels. So and also we use the in our training paradigm, we only the 3 days for the motor training, and at 4 days we make an anxiety test.
I see. It's wonderful.
Thank you.
I have another question for you.
Hello, can you hear me?
Yeah.
Thank you so much for the great presentation. My question is, in the realm of like anxiety and arousal, and I guess also related to Reza's question, were you able to pick out like a Yerkes-Dodson relationship, maybe where there's some optimal level where performance or completion of the task is best with reduction of anxiety, or was this more of like a general reduction of anxiety? Was that?
Sorry, I can't catch up your meaning. So can you?
Sure.
Yeah.
Sure. So so for a Yerkes-Dodson relationship, there's some minimal amount of arousal, anxiety. So it sort of follows this inverted bell, where at the top of this inverted bell is an optimal amount of arousal or anxiety to perform best on a given task, here exercise. But at the other end of that bell, so farthest to the right, would be extreme anxiety and arousal that would diminish task performance. I just wanted to gauge if there was a relationship with that, or were you mostly looking at reduction of anxiety in general?
You mean the motor effect on the arousal, but not only the anxiety. Well, so this is a question.
Yes.
So I think maybe it may influence may influence the arousal level, and actually what I did not mention is that actually it's not the more acceleration maybe more benefit for the effect of the alleviation of the anxiety, because the more challenge may be actually as the research mentioned, it will, and we tested, actually it will takes the arousal and the anxiety in the animals. So what we actually said is during the appropriate the challenge moment. So it may be it may be a little challenge will be benefit for the alleviation of the anxiety.
Jing-Ning, there is a question on the chat. I don't know if you can see it. They're asking, Amit is asking regarding the presence of the projections in primates, if you know if that's been studied.
Yeah, this is a very good question. So in this study, what we think is this actually the cerebellar-amygdala projections are conserved. So in the FMOST study, actually we want to test it in rats, but the the rabies, we use the different kinds of rabies virus, but we find that they did not work well in the rats. So at last we tested this effect, these projections in the mouse. But this study also suggests that these projections may be conserved in both the mice and the rats, and also the fMRI data, although it cannot provide direct evidence for the direct projections from the cerebellum to the amygdala, but we think these projection may be also conserved in the human and also maybe the non-human primates. But I think the further studies on these projections in the non-human primates will be help us to know better the function and the structure of these projections. Thank you.
Thanks for the interesting talk. I'm curious on the effect of the stress, especially peripheral effect of the stress, which is mediated via hypothalamus, pituitary gland, and cortisol. How would you differentiate these effects, which is basically independent of your canonical circuit you proposed here?
Actually, the stress can activate the hypothalamic neurons, and in other studies, but not in these studies, we actually test the cortisol level in the blood, and but not in this study. We think that in our COMS models, so this is the classical stress models, induce typical anxiety phenotype of the rats. Sorry, I can't. Did you mean I've forgotten what you asked. Differentiation between the two. What's the two situations?
Yes, you know that this stress can stimulate cortisol, and then cortisol feedback goes back to the amygdala. This is another loops which is well documented in the literature for the stress.
Yeah.
I just curious, how would you differentiate two different circuits, and how do they communicate or maybe correlate?
I see. This is also a very good question, but as I said in my talk, there are some studies focused on the interactions between the peripheral and also the peripheral and the brain. But in this study, what we think about is whether there are direct neuromechanism in the brain can mediate this motor effect on the mood. So we did not exclude the possibilities of the other reports of the cytokines or cortisol or the other factors released from the periphery, and they going to the brain to have their functions. So actually in these studies, we want to set is the cerebellum amygdala projections can also directly participate in this modulation.
Thank you very much.
Thank you. Thank you.
One more question.
Yeah, one more. Sorry. Hey, so firstly, that was absolutely wonderful. The question I have is, one of the really striking results was that a majority of those connections to the amygdala from the DCNs came from the dentate nucleus. And I was wondering if you had any reasoning or thoughts as to what would make that particular DCN particularly suited for being involved in that pathway, or if, yeah, just why do you think it might be the dentate? What makes it best for that pathway, I guess?
Sorry, I can't hear clearly. So, but I guess you ask the density of the projections from the DN to the amygdala. Is that right?
Yeah. Yeah, I was asking, is there anything in particular about the dentate nucleus that you think makes it suited for being involved in this pathway?
Yeah. This is also a very good question, because actually in the three cerebellar nuclei, the dentate nuclei is emerges in the newest in the evolution. So its connections is mostly to the cerebral cortex. So it may be mediated more high functions of the cerebellum. So our findings that the cerebellar dentate nucleus projection to the amygdala actually is in line with these notions. So we also think the function of the function of the actually the cerebellum to modulate the emotion, the emotion is the requirement, maybe the requirement of the evolution. So these projections may provide more provide a mechanism for the brain to coordinate the motor and the motor responses and the emotional responses to generate a more appropriate responses to the changes of the environment.
Thank you so much, Jing-Ning. That was amazing, and congratulations on really outstanding work. And thank you for your time in teaching us today.
Yeah, thank you, Risa, very much. So I just want to say sorry about my poor English. Maybe I can not catch your meanings correctly. But I hope my answers maybe provide some information you are interested in. So lastly, thank you. Also, thank you very much. Thank you very much for your listening. Thank you, Riza.
No, my pleasure. And your science speaks for itself.
Thank you. Thank you.
See you later. Bye-bye. Bye, everyone.
Bye. Bye. Bye.